Thank you for joining. This is great. Really good turnout for the inaugural or first seminar of the season. I think we're cracking the code for getting people to attend, moving it to midday, providing some food. My name is Benjamin Smith. I'm the director of CORE, and I'm not going to introduce our speaker because we have a student, Paolo Mallo, who's going to introduce the speaker. But I just wanted to thank all of you for being here. Please open up those emails from Core, see what's going on. We're doing a lot of interesting stuff that I think is of broad interest to whatever program you might belong to. So thanks again, and we'll see you around.
Hi, everyone. My name is Paulo. I'm a medical graduate trainee in Brazil, and I'm a new student, a master's degree student here from CORE. And it is my pleasure to introduce Dr. Nandini Dendukuri to you. Dr. Dendukuri is an associate professor at Department of Medicine, McGill University, Montreal, and associate director of CORE. Her research focuses on developing Bayesian statistical methods for diagnostic test evaluation in the absence of a perfect reference test. Is supported by grants from CIHR and CERC and NIH. She's currently an editor with the Crochet Collaboration's Diagnostic Test Accuracy Evaluation Group. So please. Uh, thank you. Thank you very much, Paula. Do I have to use this mic? Okay. Because usually people have to tone down my volume without the mic, but I will use the mic here. Thank you all for being here. Delighted to have this big turnout. I was actually a bit concerned that the title would turn people away. So I'm glad maybe it's the sandwiches, but I'm happy to see you here. Um, so as the title says here, I'm going to talk about whether when prevalence and diagnostic accuracy appear connected, is that bias or is it biology? This question actually came to my attention a few years ago. I mean, as is typical with research, your current research project paves the way for the next one. And I was doing a project with a veterinary scientist at Saint Hyacinthe. And in veterinary science studies, they often will analyze, they will often evaluate a test in different herds or in different groups, somehow different populations. And my co-author, Sebastian Bugzinski of University of Montreal, he had a line in the discussion which caught my attention. We finished off saying future studies need to look at whether, at how and why prevalence informs sensitivity. And I thought, why? Prevalence and sensitivity have nothing to do with each other. But he said, no, you see, there's this paper published, and he showed me a paper by some influential authors I'll just share with you. And that basically led me down this rabbit hole. And here, this work is done jointly with Yang Lu, my PhD student. Yang, I hope you're online because I've borrowed a lot of his slides. And Robert Platt, who many of you know, is a now director of School of Public Health, is it? At maybe. All right. So I'm going to begin by going over what prevalence and diagnostic test accuracy are. You're familiar with the concepts, I'm sure, in this audience, but still, I'd like to underline to you the difference between the theoretical concepts and some empirical evidence that has appeared in the literature, which seems to be somewhat different from the theory. And I'm then going to explain to you how in this project we looked at the impact of bias on that empirical evidence that one sees. Why is there a difference between the theory and the empirical evidence? We're going to explore that using directed acyclic graphs and simulation studies. Then I'll finish off with a discussion. So this, by the way, forgive me, I'm working on a Mac, and this computer we have here is a PC. So you have a mix of Courier and Calibri fonts. Uh, all right. Now here you see this table. Yeah, this table, you all know, this is what you see in Epidemiology 101 in the chapter where you study diagnostic test accuracy. You've got some diagnostic test whose sensitivity and specificity you're interested to study, and you compare that with what we now increasingly refer to as the target condition. So this is terminology coming out of Cochrane collaboration. Uh, and the test is often referred to as the index test, the test under evaluation. And with this 2 by 2 table, you can obtain estimates of the parameters that interest us. The parameters that interest us are the prevalence of the target condition, the sensitivity and the specificity of the diagnostic test. And as you all know, parameters are theoretical concepts. They are defined in the population, but we seldom get to observe the parameter itself. We typically draw a sample from a population and use a sample to obtain estimates. So if you have a 2 by 2 table like this, you can calculate these simple proportions to obtain estimates. And you'll see I'm using this hat notation over here, which distinguishes the estimate from the parameter. So pi is the parameter I want to know about, and pi hat is the estimate that I can get in my sample. And you'll notice that in calculating pi, I'm going to basically make use of all the cells of the table, but in estimating sensitivity, I'll only be using the first column where the target condition is positive, and for the specificity, I'll only be using the column where the target condition is negative. And this is why you expect that prevalence has nothing to do with sensitivity and specificity. So by definition, these parameters, sensitivity and specificity, are expected to be completely independent. So if you're a clinician, patient comes into your clinic, you carry out a diagnostic test on them, and the result of the test should have nothing to do with how many people in the population have that condition. It's the characteristic of the test. Besides sensitivity and specificity, we also often calculate predictive values. So you can use that same 2 by 2 table to obtain uh, parameters that are actually of far more relevance in a clinical setting. You want to know if a test is positive, what's the chances that the subject has the target condition? And same for when the test is negative. And these expressions here, oh, sorry, I've got a typo. This should be NPV for negative predictive value. But these expressions for positive and negative predictive value, you'll notice they are functions of the prevalence and the sensitivity and the specificity, which I had defined on the previous slide. So by the very definition, you expect these quantities to be associated with the prevalence. And because of that, when you're interested to evaluate a diagnostic test, you've done your individual studies, you want to do a meta-analysis now, you want to collate information across different studies to see how well this test performed in different settings, we tend to focus on the sensitivity and specificity. So every study in our meta-analysis might will have a 2 by 2 table, and we'll be able to get both the sensitivity and specificity as well as the predictive values, but we don't pool the predictive values in a meta-analysis because we expect that the predictive values will vary across studies. So that's not what's interesting. What is interesting to us is the sensitivity and specificity, because this is what is going to allow you to say, is my test good for use in all settings? I'm going to illustrate a bit, though, the contrast between theory and what we sometimes observe in practice using some examples. Here's a study I was once part of about 10 years ago. It was a meta-analysis of the gene expert test for tuberculosis. And this test was a game changer for TB diagnosis. It's a rapid point of care nucleic acid amplification test. And uh, we looked at this test ad nauseum. Ian will confirm. We've done so many reviews and updates and so on of this, but I'm going to talk to you about two particular cases, the case of pulmonary TB and extrapulmonary TB. And the reference test for both of these studies was bacterial culture, and these reviews were published in the Cochrane collaboration carried out by some excellent researchers, very diligent research. And here's an excerpt from the 2014 review on pulmonary TB. This is the forest plot in the meta-analysis. So unlike in randomized controlled trial meta-analyses, you've got two parameters of interest now. You've got sensitivity and specificity. So, typically if you're pooling data across RCTs, you've got a risk ratio or a risk difference or something like that. Here you've got two parameters to deal with, sensitivity and specificity. And this is what these forest plots illustrate, how these values varied across about 40 studies over here. And just quickly, it seems like the specificity is quite high with respect to culture, and sensitivity. There's some variation in sensitivity over here. And if you line up if you line up sensitivity and specificity versus prevalence, as I'm doing in these scatter plots. So the scatter plot on the left is sensitivity versus prevalence. The scatter plot on the right is specificity versus prevalence. And all I'm doing is I'm just taking these, the point estimates of sensitivity and specificity across these studies, and I'm calculating an estimated prevalence over here. And I'm using the term prevalence kind of loosely. It's not the prevalence of TB in the population from where these studies were carried out, but if, for example, it would have been the proportion of TB subjects in the clinic where the individuals were recruited from. And here you'll see in red are lowest plots. So not best fitting straight lines, but best fitting lines, curved lines if necessary, which allow us to see what's the possible relationship here between these variables on the X and Y axes. So what you see is that sensitivity does indeed seem to be unaffected by prevalence. This is what we'd expect. This is what I was saying to you at the beginning, that our theoretical understanding of sensitivity means it should not be affected by prevalence. But the specificity seems to be kind of curving downward. It seems that as prevalence increases, specificity is going down somewhat over here. And any suggestions from anybody why this may be happening? Yes, yes. Yeah, yeah. Yes. Yeah. Well, see, very helpful to have PB experts in the audience, huh? To answer questions like this. So, I'm sorry, before I go to my own explanation, I just want to share this slide. This is what happens to the predicted values. So here you have positive predictive value versus prevalence, negative predictive value versus prevalence. And as we expect, these graphs show that these parameters are affected by the prevalence. Positive predictive value will increase, negative predictive value will go down with prevalence. This is what we expect. So indeed, in the case of pulmonary TB, it's possible that there is some confounding. And it's true that in some of the studies in that meta-analysis, there were more subjects that had a history of TB. And when subjects have a history of TB, there's a chance that that nucleic acid amplification test is giving you a false positive. But also, in general, you'll find that it seems to affect the estimate of the prevalence. So it's possible that we slightly overestimate the prevalence in those studies and we slightly underestimate the specificity, and that may be causing that apparent negative relationship that I showed. And but that this is conjecture. This is the hypothesis. This may be happening. We don't know for sure. Empirical data here. Here's another example of this time on lymph node TB. And once again, I'm looking at the relationship between sensitivity and prevalence in this graph on the left, and specificity and prevalence on the right. And this time we see a different result. This time we see that the sensitivity seems to be changing as prevalence increases. This is surprising, but sensitivity seems to be increasing. Specificity, on the other hand, seems not to change much. Is there a slight increase here, perhaps at the end? But seems less affected. I mean, this is stark. So again, any suggestions on why this may be happening? Yeah, yeah. Mhm. Right, right. Yeah, right. Yeah, yeah. So, yep, good answer, Kaveri. That's at least a part of the answer. So, yes. And here again, I just want to share with you that the predicted values will vary with prevalence as expected. Uh, So, yes, what's happening here, perhaps, and again, it's a hypothesis that it may be reference standard error bias. Bacterial culture doesn't do very well in picking up lymph node TB. It has suboptimal sensitivity, but near perfect specificity. And what you're talking about, Kaveri, spectrum, may be caused by varying bacterial load across subjects in a study. So those subjects who have a high bacterial load, a more severe case of TB, are likely to be picked up by both the index test and the reference standard. And in the case when a subject has a low bacterial load, it's possible both tests miss that case. So all this put together, we'd expect that there's possibly an underestimation of TB prevalence because culture's got suboptimal sensitivity. It's not picking up all the cases. So our so-called estimate of TB from these studies may be low, and our gene expert specificity may also be underestimated over here. But it's possible that we're overestimating gene expert sensitivity. Why? Both tests do poorly, but because we are relying on culture to tell us how good gene expert is, culture is not picking up the errors gene expert is making because it's making the same errors. Yes. Yeah. Yeah, yeah,
And
you can ask.
so earlier you said the prevalence on the plots is sort of at the clinic, but is it is the prevalence in the plot just amongst the participants estimated from
Yes,
participants, or is
yes,
it estimated?
correct. Yeah,
Okay.
that is correct. Yeah, I'm not using prevalence in the proper public health usage of it. I mean, really, the proportion of people who have TB in that particular study. And, all right, so I don't know why we have a blank slide. I hope, but let's hope nothing is missing. Yes, what I want to share with you is that these observations have been made before, and indeed, this was the study that my vet science colleague pointed me to. In the year 2009, in Journal of ClinEpi, uh, Mariska Lieflang, Patrick Bosaut, and Les Zerwig, all people active in diagnostic test evaluation methods, wrote this paper saying that diagnostic test accuracy may vary with prevalence, and they discuss the implications for evidence-based diagnosis. And in this paper, they did a review of the literature, and they found that they looked at various conditions, dementia, I guess multiple sclerosis, UTI. And mainly what I want to draw your attention to is these arrows here. They looked at how prevalence varies with sensitivity and specificity. And, sometimes it was up and sometimes it was down. And there wasn't a clear, pattern. But there does seem to be some pattern. And in the two simple examples, like I showed you, there does seem to be a pattern, but it's not always the same pattern. And here's another study. This is an excerpt from CMAJ published in 2023. CMAJ somehow loves this topic. I don't know why, but they published a few articles on this. And this study, which actually I'm still a bit perplexed by what it means. But here they took 6,909 studies of diagnostic test accuracy. So they got a whole lot of studies out of the Cochrane database, and they just threw them into a statistical model. And in this model, they found that overall, across all these studies of many different disease indices and many types of tests, in general, sensitivity seems to be increasing with prevalence and specificity seems to be decreasing with prevalence. And I don't know. I'm not sure what to make of that. But anyway, it's there and you get published in the CMA like this by analyzing lots of data. So essentially, there seems to be some uncertainty in the literature. What are these patterns? Why do they arise? And really, in order to properly study that, my student Young and I, we felt that we need to take a theoretical approach. We need to first understand what biases may be coming into play and use simulations to isolate these biases. Because all of the studies we're looking at, which are in the published literature, we don't know. There may be multiple biases underlying each data set. It's hard to know, is it the history of TB? Was it perhaps reference standard bias or some mix of things? So we need to take this, look at this problem from a theoretical point of view. And whereas DAGs are used a lot in etiological studies, they've not been used in diagnostic test accuracy studies. So that gave us an opportunity to explore the usage of DAGs. And actually, it was a talk here. James Brophy gave a talk here on DAGs and how he found them illuminating. And actually, that's what set me off. I said, okay, let me see what I can learn from DAGs. And it was not easy. I couldn't see anything from there for a while. But anyway, just a few words on what is a DAG. DAGs are useful for illustrating causal relationships between two components, two variables. And sorry, yeah, I think I've mixed up my terms here. But they're useful for illustrating causal relationships between two variables. And the components of a DAG are the vertices and the edges of the DAG. So the vertices are variables and edges are arrows connecting the variables, and the arrows depict the causal relation. A characteristic feature of a DAG is that there is no closed cycle. And if you look at the, etiological study literature, you'll find several articles on DAGs, and there are well-recognized patterns that describe confounding, misclassification bias, selection bias. All of biases that you, again, study in Epidemiology 101 are well characterized in etiological studies. So, but in diagnostic test accuracy studies, the variables that interest us are the target condition, and that target condition may be observed or it may be latent. So in the case of lymph node TB, for example, it's, we're trying to get to it by using the culture test, but the culture test is a poor measure of lymph node TB. In the case of the pulmonary TB, I think the culture test is considered a pretty good test, and therefore it does a fairly good job of capturing the target condition. So sometimes you are able to observe it, sometimes not. The index test is the test under evaluation. And besides these two variables, there may be covariates like the history of disease or the severity of disease. Other things may come into play. There may also be selection criteria that affect diagnostic test accuracy studies. So, for example, if you're evaluating a screening test, then you will often see designs where all subjects were put through the screening test, but only a greater proportion of those who are positive and a smaller proportion of those who are negative go on to receive the reference test. So, selection criteria may also come into play in creating a DAG, and this edge is what we want. We want to get after the sensitivity or specificity test. What's the time? Third. All right. So reference standard error bias. I'm going to basically go through a series of DAGs that lay out the different kinds of biases one commonly encounters in these studies. This is how we would depict reference standard error bias in a DAG. There's a target condition. The target condition affects the reference standard, and it also affects the index test. And I'm just going to jump ahead here. What we want actually is to study the association between the target condition and the index test. But if the target condition is not easy to observe, then we're going to rely on some reference standard. And in fact, the association that we're able to study is this red line connecting the reference standard to the index test. We're not able to study just this green line. We've got this target condition here coming into the middle of the association that we are going to study. And I like to read it this way. I'm not 100% sure the correct thing to do, but I feel that the association between the reference standard and the index test is affected by that target condition or the prevalence. So in other words, the sensitivity and specificity that we're going to estimate from these studies, those estimates can be functions of the prevalence of the target condition. Indeed, what I'm trying to say to you is, I personally didn't find that just looking at the DAG, turns on a light bulb and say, oh, that's the bias, that's the source of the bias. So I'm trying to interpret it like that. But finally, as I'll show you, the simulation studies, the probability equations were finally what made a difference for me. And maybe I'm just full bash, but there, yeah, that's yes, right. And somehow I feel like this red line here, this thing here in the middle, can influence it, is how I read it. But yes, I'll set that aside, and I'm going to just show you some simulations that we looked at. Essentially, Young looked at what would happen if the reference standard was perfect. That's we're going to see a diagram where you have purple crosses indicating when the reference standard is perfect. And he looked at varying values of the sensitivity and specificity of the reference test. The test under evaluation had 90% sensitivity and specificity. And we looked at different values of prevalence, because what we're trying to capture is how does change in prevalence affect change in the estimates of the test under evaluation. Yeah. With the target condition. Correct. That's right. Yeah. That's the. So, yeah, Ben is.
Sorry, I just asked for clarification as to whether reference standard being perfect was in reference to the target condition or to its performance as a diagnostic test. And the answer was to the target condition.
Yeah. So here's what we see. That those purple crosses are essentially all, they're sort of jittered. They're just randomly scattered around the 90% true value. Sorry, on the y-axis, you've got the estimated sensitivity. On the x-axis, the estimated prevalence. So as prevalence is varying, you see that when there's a perfect reference test, there's no change. But the moment you have a less than perfect reference test, you start seeing that increasing relationship. So maybe that CMAJ analysis that I just showed you, perhaps it suggests that many times dealing with imperfect standards. Wow.
So here you didn't allow for the possibility that the diagnostic test might in fact be better than the reference standard,
Yes,
right?
absolutely. You're right. That's correct. And we'll look at that in a moment. Correct. Okay, you got it. Good. I have all these good students over here anticipating the next slide. But yeah, here again, when you have perfect reference standard, no problem. You don't see that anomalous pattern. This is the relationship between specificity on the y-axis and prevalence on the x-axis. But the moment you have an imperfect reference standard, you see a decreasing association. And again, we have the luxury here of looking at simulated data. So we are able to isolate the bias. It's not mixed up with other biases. Here's reference standard bias plus what we call misassumption of conditional dependence. So what happens if there's another variable? And in our example, that would be, let's say, bacterial culture, in the example I shared earlier. Sorry, not culture. It would be bacterial load. So an individual might have TB, which is what we're trying to pick up with GeneXpert and the bacterial culture, the reference standard. But both the reference standard and the index test may be affected by another variable besides the target condition. And that could be, in this example, bacterial load. So both of them would do poorly or very well, depending on that. And so when you have that additional variable involved, again, I'm just going to skip this. These are the simulation settings. But here, we no longer have the perfect test case because you can't have dependence if you have a perfect test. To have that, behavior where the test's accuracy depends on another variable, that means the test is not perfect. And so once again, you see these associations between sensitivity and specificity and prevalence. Another commonly recognized bias in diagnostic test accuracy studies is called spectrum effect, or also sometimes called spectrum bias. And here is a situation where that additional variable R affects the index test only, not the reference standard. And again, our simulations show that if you have spectrum effect, because indeed, you've got maybe individuals who have, I'm trying to think of a good, like the history variable that we talked about earlier. If there are different, if some individuals have a history of TB and others don't, and that history of TB is affecting the performance of the nucleic acid amplification test, but not the culture test, then you would have this scenario where, again, you can show that there is going to be a variation in sensitivity and specificity with prevalence. And here's another type of bias where that extra variable also affects the target condition. So this is kind of obvious. It affects the prevalence and the target condition. And this is exactly the same kind of DAG one would see to depict confounding in an etiologic study. Here, you have actually one situation where you even have a negative association between sensitivity and the prevalence. I mean, it was typically a positive association, but here you have a negative association. And yeah, that's right. And that is despite the test being perfect. So even if you have a perfect reference test, confounding can create this association between prevalence and test accuracy. And finally, this is how one would depict what we call verification bias. So a selection bias where the index test, perhaps a screening test, is used to determine who goes forward. So V is a verification indicator, and only those who have V is equal to 1, say, go on to receive the reference standard. And again, drawing a DAG like this for me didn't straight away say, oh, here is the problem. This is a DAG, I can tell you. We've thought through it carefully in this. The DAG is correct, but we had to do simulations before I could understand what's happening. And here again, one sees these patterns. And then what happens, I guess one of the main reasons I'm interested in all this is we have methods, statistical methods that would correct for these biases. You can correct for reference standard bias. You can carry out subgroup analyses or regression analyses to correct for covariates that you've observed that are affecting test accuracy and prevalence, that are causing confounding. But people don't use it. So if you look at your typical Cochrane collaborations, diagnostic test accuracy, meta-analysis, and systematic review, they'll have this quadus diagram, which shows all the risk of bias in every study. And they do not even, sometimes they recognize there's reference standard bias. Sometimes they don't even recognize it. But even if they do recognize, we just document. We document and say there is reference standard bias, and that's it. I mean, we don't try to correct for that bias because, clearly the main problem is that the estimates of sensitivity and specificity that are coming out of these studies are not accurate. They're biased, and we really need to be adjusting using these methods that we have at our disposal. So in summary, what we found is that reference standard bias, error bias is especially problematic and result in a spurious association. But things like spectrum effect or confounding, those are genuine relationships. And what you want to do in the presence of these kinds of problems is to carry out subgroup analyses. So if you know a certain variable is causing confounding, do what you do in other kinds of research. You carry out a subgroup. If you look within categories of individuals who had a history of TB and didn't have a history of TB, then you'd be able to get a better estimate of the sensitivity of the test. And like I was just saying, the biases are routinely recognized using tools such as Quadus, but not always adjusted for. And I'll just leave you actually with a word from that original paper by Li Flung, which was written with an eye on providing some guidance for how all this should influence diagnosis in a clinical setting. And so, as I say here, sensitivity and specificity are not fixed test characteristics, but test properties that describe the behavior of the test in a particular situation. As the setting, filter, or patient group changes, prevalence and accuracy estimates may change. So I think one of the problems is that they refer to prevalence and test accuracy without distinguishing between parameter and estimate. And the problems that arise, the things that are, the patterns that are confusing arise when we're looking at the estimates, which is, after all we have at our disposal. We don't see the parameters. And here, for this reason, variation in disease prevalence and test accuracy can act as a flag for readers to detect differences in study population or study design. I think that line is relevant for researchers, especially. If you're doing a diagnostic test accuracy meta-analysis, take a look at whether prevalence is associated with your sensitivity and specificity estimates, and it's a flag to perhaps carry out a suitable adjustment. And in terms of for clinicians, as they say, clinicians are advised to base their decisions on studies that closely match their own clinical situation, using prevalence as a guide to uh, using prevalence to guide the detection of differences. So I suppose if you come across a diagnostic test accuracy study with a prevalence that seems similar to what you would encounter, then its estimates perhaps make sense. But if if you're going to take a very different prevalence, then you know perhaps it's not relevant to your situation. Those because the sensitivity may be very different in that setting. And yeah, I mean, so this paper, as much as I think it's a fantastic paper, and it's written with they have a great intuition about the problem, but it's written in very general terms. And I think that there is a need to sort of refine and be more precise about why these biases are happening. What are they? Like I said, study the biases in isolation to understand how we can do better to analyze and interpret these studies. I think that's it. That's my last slide, some bibliography. Thank you.
Um, so please, anybody that's watching us on Zoom, please leave the question on the comment section. If you leave it, I will read it out loud. Okay, thank you. Any questions here on the room? Okay, please.
I think part of the problem that was great, Nandini. It's always nice to go back to FE 101 and Stats 101, which is probably where I always land before I think of anything else. But I think part of the problem is the people who develop diagnostic tests are not necessarily epidemiologists and biostatisticians, right? So they'll put something on. And I think most of the tests are with enriched artificial populations, where they'll say, give me 50% with the condition, 50% with. So they're never based on a population. And I'd even argue the bacterial load issue is an example of spectrum bias because we have different loads of microbes, right? So it's not just the test. So I'm not even sure how one would go about starting the process of adjusting for the issues that you raise, because I suspect they're quite variable.
That's right. So indeed, it's maybe that's the reason why people aren't attempting to do it. It's challenging, and sometimes you can recognize the source of the bias. I mean, the variable that's causing the spectrum effect is a measured variable, but many times it's a latent variable. But then again, at least you can do a sensitivity analysis, saying what if, what if I had access and the variable had a certain distribution. But yes, you're right. It's challenging, and maybe to be complete at best, one can carry out a sensitivity analysis and say, what if I were able to measure another variable that explained that impacted prevalence and sensitivity.
Yeah, thanks again, Nandini. Really perfect talk. Broad interest, I think, at least to me. I share your opinion about DAGs versus just simulations, that I appreciate the intellectual exercise, and I know it's informative to some, but I always need to verify it with the simulation. So following that up, actually, for the simulations you did, are you able to sort of validate what you observed using real world data? Maybe like quantiferon. I imagine there's been tons and tons of published studies where maybe the populations varied in different ways to sort of show that indeed such sort of findings are observed in real world. I know the CMAJ was sort of this, sounded like just a brute force, like there's a rough trend related to prevalence, but maybe dissecting the different.
I think that would be worthwhile. And there is, I think, a table in that paper that attempts to do that. But yes, I think that it would make a lot more sense. I think it would be a lot more interesting to break it down and see, which test and which population, and understand whether we can replicate these patterns in real life. To what extent? Thank you. Yeah.
This is the row of nerds, I guess, but I'll also really, really appreciate that talk. What's the mechanism, I guess, for the association of prevalence with these?
Yes.
And yeah, does it shed light onto the mechanism, or is it purely sort of something mathematical?
Right. You can actually, write down equations that are, we don't have a board here, but, you can, that 2 by 2 table, you can write down mathematical equations that relate each cell in the 2 by 2 table to the prevalence and the sensitivity and specificity of both the reference test and the index test. And so you can, and that problem is actually what we call a non-identifiable problem. You've got 4 cells in the table, and 4 cells in the table means you have 3 degrees of freedom. You can estimate from that problem some 3 parameters only. But I just enumerated for you 5 parameters: prevalence and the sensitivity and specificity of each test. So to really study anything from this data, you need to fix something. So what we do is fix the reference test to have perfect sensitivity and specificity. That's our default approach, because that's the easy thing to do. But let's say, okay, we don't want to do that, and we're at least willing to say, okay, sensitivity of the index and sensitivity and specificity of the reference test, I fix them at some values. I say bacterial culture has 0.95 sensitivity and 0.99 specificity. Now, then I have essentially 3 equations and 3 unknowns. So, if you go back to your high school days, remember we first studied simple equations, 1 unknown, 1 equation. Simultaneous equations, 2 unknowns, 2 equations. So what I'm just describing to you, 3 unknowns, 3 equations. And then you can use that to then characterize how the specificity and sensitivity estimates of the index test are varying, with the prevalence and with your guess values for the reference test sensitivity and specificity. So you can actually derive mathematical expressions and see, are you underestimating? Are you overestimating? Does that answer your question?
Yeah, so it's really
okay. Yes, exactly. That's right. So behind those simulations are maths, but then today we can, simulations will just allow us to do so much more.
Relate to the other work that you've done where you use,
well, thank
3
you for the
tests.
question. And indeed, like I was, alluding to earlier, when it's actually through work like that when someone tells me things like sensitivity is going to change with prevalence, no, it must be reference standard bias, and you are resistant to adjusting for it. That's my immediate reaction. But it's yes, reference standard bias is a big part of it, but it's not the only source of bias that causes that. And yes, how does it relate to my other work? It's because I want to underline that there are methods that one can use to adjust, at least as a sensitivity analysis. If not your primary analysis, at least as a sensitivity analysis, you can ask, what if I acknowledge my reference test is not perfect? How would it change my conclusions? And in fact, in that Cochrane review of lymph node TB, we actually did do that. We did do that sensitivity analysis. And Cochrane, I don't know if And if you've worked with them, they're very regimented. They have their way of doing things, and you can't stray off that path. Even if it's wrong, doesn't matter. You got to do it the way they wrote it down. And one of the things they were not so open to doing is Bayesian analyses or adjustment for a imperfect reference standard. But I mean, of course, they were open to it once we discussed, and yeah, we got to apply these methods. So yes, that is how this is connected.
Hi. Please tell me if you disagree with this assessment, but in your modeling examples with the imperfect tests, it
Yeah.
seemed like sensitivity was rising logarithmically with prevalence.
Okay.
So considering that, I was wondering if you think some level of thresholding where a change in prevalence at the low end of the spectrum would be more meaningful versus a change in prevalence at the high end might have a negligible impact on sensitivity.
Yes, and maybe that is what I left you with the line from the paper by Lee Flung. They were saying if you're trying to if you have a particular study with different prevalences, then maybe if you focus on those where the prevalence is similar to your own situation, then you can apply the sensitivity and specificity estimates from that type of study. I mean, I'm not fully answering your question. I mean, the exact nature of the relationship, frankly, I'm not sure what's the mathematical form. What you're saying is that at lower prevalences, it seems to be changing fast, and then it sort of plateaus at higher prevalences. But those were some settings that I showed you. Maybe if you change the settings slightly, you'd see a slightly different graph. So I don't think we can generalize to all settings, but I think this much is clear. Yeah, you might want to look at those studies where the prevalence is actually in keeping a situation to which you're trying to apply the results of the study.
Thanks.
Any other questions on Zoom?
No food for them. Yeah. Thank you all.